Hello and welcome to another episode of the Omics cast. Today we're going to talk about liquid biopsy and NGS. As a reminder, if you want to follow me on Twitter, you can follow me on twitter.com/Alberto. And if you want to contact me via LinkedIn, you can also do so. I'm happy to answer any questions you have. And a reminder that this slide deck is available upon request. Today we're going to talk about liquid biopsy and NGS and what's been happening lately and what is roughly what this field is currently today and where it is evolving into. Liquid biopsy is a promising new field to improve detection of disease conditions with non-invasive sampling, and combined with NGS, which gives you very high resolution data, it can be used as an underlying tool to detect these disease conditions very precisely. So how does liquid biopsy compare to a standard biopsy? A standard biopsy is usually a time-intensive procedure, and it has to be done by someone highly trained. It localizes the sample in a given tissue or part of the body, which is usually not easily obtained. And it's thus invasive, and importantly, it has some pain or risk associated to it. So every time you take a biopsy, especially certain parts of the body, there's a risk associated that you're gonna injure the patient, right? The patient that you're trying to diagnose. Liquid biopsy, on the other hand, is very quick, and it gives a comprehensive view of, in this case, what's in blood, and we'll see also DNA within blood, what is in there. It can be easily obtained, minimal pain and risk, and it's minimally invasive. What has happened and why do we care about liquid biopsy? What has happened in the last 50, 60 years? In 1948, it was discovered that there is circulating DNA in blood. And this could not have been the case. And biology keeps telling us that we're not designed intelligently, but rather we drift through evolution. And so the fact that we have DNA circulating in our bloodstream is a happenstance that could have happened, could have not happened, right? But it is important today for us to be able to do diagnostics on disease conditions. In 1977, detection of high concentration of cfDNA in individuals with cancer. So in some stages of cancer, the amount of cfDNA, the concentration of cfDNA in blood increases. And that can be in itself already a way of stratifying individuals. Then there's been some other advances in what you can detect in cfDNA. And in 1977, it was shown that there is also fetal circulating DNA in pregnant women. And 1997, and this opened up the field of what's been dubbed NIPT, or non-invasive prenatal testing. Later on, there were some more advances in starting to apply that, and as you can see, in the last, I would say, 20 years, there's been multiple efforts in applying the knowledge that is accumulating on cfDNA to detect disease, and many of them are around cancer. One aspect of what's important to understand in cancer is what's called tissue clonality, which is can we find out how the different metastatic loci are evolving or have evolved in the progression of cancer. And this is a depiction here of shown in different colors, different subclones of cancer, each of which can have different accumulating mutations of somatic mutations with respect to the individual's DNA. And then if you take tissue biopsies here shown in this what they call bread loaf sections, then you can find out about the different clones in this tumor, right? What's important is that to some extent, and we'll see some of these later on, you can also replicate this same approach of finding the different clones as shown here in tissue biopsies. You can also replicate it to some extent in liquid biopsies. And so something that is important to realize, it gives an option for doing the same in liquid biopsy. In terms of sensitivity and where can liquid biopsy be applied, here there's some techniques, direct sequencing, parasequencing, PCR-based, and at the bottom, digital PCR, which many people claim is the most sensitive assay possible. Liquid biopsy really has opened up as a field as we've started having available these assays that have very high sensitivity, because they can find the needle in the haystack amongst all the DNA circulating in blood, those molecules of DNA that one is interested in finding, right? So digital PCR and NGS here, next generation sequencing, have opened up the field tremendously for liquid biopsy. And so, in a sense, what is liquid biopsy? Well, it's finding, here we're starting with DNA, finding something in the blood that is predictive of something. And on the left-hand side, we have the case for monitoring pregnancy due to the fact that the fetus sheds DNA, the DNA of the fetus, which is half the DNA of the mother and half the father, sheds DNA into the bloodstream, and then if you take a sample of the blood of the mother, you'll find both maternal DNA and fetal DNA. And the fetal fraction increases as the pregnancy progresses. And so if there's something wrong in the DNA of the fetus, you can identify it through NIPT, non-invasive prenatal testing. Liquid biopsy applied to cancer is similar in concept, meaning if there's a breast tumor or a lung metastasis or a liver metastasis, these different clones here in different colors will shed DNA into the bloodstream, and if you're able to detect that tumoral DNA within the total fraction of the DNA in blood, then you'll have a way of detecting cancer and cancer progression, right? And so the fraction of this tumoral DNA in blood increases as the stage of cancer progresses. So what are important considerations when looking at liquid biopsy? The first important consideration is what is inside, what is in the blood, what is inside the sample. Importantly, there is cfDNA, and most of the work that's been done in liquid biopsy focuses on this part. There are also red blood cells, which in humans don't have nuclei inside. There's platelets, plasma white blood cells, exosomes. So these exosomes can also contain DNA, and there's different hypotheses on why they are there and what they contain. And there's been work done to find out if they have a prognostic, a diagnostic value. And then there's CTCs, cells that are circulating that can be tumoral. Also microRNA. So of all of these, we're going to talk about a little bit about CTCs, but also mostly cfDNA. In terms of considerations before the analysis, what do you do from the blood sample that you've drawn? First of all, you have to put it in a tube, and the type of tubes and the coating in the wall of the tube is important, and it is an important consideration for stability and the purity of the DNA that you can get at the end. And then you need to spin it, and this is done by a double centrifugation step, first centrifugation, second centrifugation. And then you've separated from the cells, the buffy coat, to the rest, which is a plasma and contains the cfDNA. And then you have to extract and purify the DNA inside that plasma, centrifuge plasma. And there's also different ways of doing it. An important consideration is how do these scale in terms of doing it on a liquid handler, meaning you can do it manually, you can do a handful a day, but can you do the same process, the same quality with a liquid handler and do hundreds a day? Once you got it extracted and purified, you need to quantify it, and there's different methods here. The ones that are in this slide are a bioanalyzer, DNA quantification, nanodrop, or a picogreen DS assay for double-stranded DNA assay. Then you can store it either in liquid nitrogen or -80, -20. There's plenty of work having done this at -20 that shows the validity. A lot of people prefer to do it at -80. And then you detect whatever's inside the cfDNA in different techniques. And we're going to touch upon digital PCR and then show some data, some results on NGS. So very quickly, circulating tumor cells for someone that's got cancer, not only the ctDNA for tumoral DNA, but also the CTCs can be important in diagnosing that cancer. For the DNA, you can do what you usually can do with DNA, which is find variants, insertional deletions, epigenetic marks, which I'm not going to talk in this slide. Chromosome aberrations in this talk. I'm not going to talk about epigenetic marks, chromosome aberrations, etc., etc. All that we know you can do with DNA. With the cells, you can do more, and you can treat those cells with, I would say, longer, more complex procedures, and those will give you. There's a little bit of summary here. They can give you a more fine-grained, detailed information of what's happening, not only in terms of the tumoral condition, but also inflammation state of the individual. And something that epitomizes this is a single-cell omics, in this case, single-cell RNA-seq in this slide, where the circulating tumor cells are put through a microfluidic device that isolates them one by one, and then you apply a high-throughput omics assay, in this case RNA-seq, that gives you these clouds of the different cell populations, the heterogeneity in CTCs. And this can be very important to really understand what the cancer is doing in that individual. In terms of the DNA, these are two different versions of similar depictions. The DNA goes into the bloodstream through different methods, and importantly, those methods can affect or alter the way the DNA goes into the bloodstream and how long it lasts, how short or long the molecules are. And here it describes apoptosis, necrosis, and secretion. And secretion is an important one. There's more and more discoveries being done around secretion of DNA linked to cancer. And then once you have it in the bloodstream and you've centrifuged it and you have your plasma or serum sample containing the potentially tumoral DNA, then you can do your NGS or your ddPCR. It's important to know where this DNA comes from, because it can tell you more about if this DNA belongs to a healthy cell of a healthy nucleus, or a diseased cell, a tumorigenic cell. And the reason for this is because DNA is usually wrapped around in nucleosomes like this. It can also be opened up for transcription or to do something with it. But lots of the DNA that we have in the bloodstream has been there as a nucleosome. It can be as a mononucleosome or as a dinucleosome with a linker region in between. And depending on the method of shedding this DNA out, as shown here in the previous slide, apoptosis, necrosis, secretion, etc., the average length of the histogram of the length of these cell-free DNA fragments will change. And there is plenty of papers to show that in healthy volunteers, there's a peak at around 165, 166. But in cancer patients, this DNA length can narrow down, can shorten up, and have these different peaks at shorter averages than in healthy volunteers. And so this altogether has created a subfield of this liquid biopsy field in which people try to enrich for these shorter molecules that are that have the potential to be cancer molecules. Now, touching on ddPCR, this is yet another collection of methods that has developed through microfluidics. And here there's three of the possibly main players, Bio-Rad, Raindance, and Stilla. There's more now, where they all offer these digital droplet PCR methods, which many of them are targeted, are specifically designed for detecting cancer in blood. And so what you essentially do is you put your DNA in a tube, and this tube has these beads or other ways of attaching the DNA to a physical molecule, and they will shed light different frequencies with different fluorophores depending on the DNA being a wild type or a mutant, right? And so once you put these bits one by one into a machine that detects the light, then you can get a cloud of the different dots and different colors. And so all of this is through microfluidics, which basically create these micro droplets, out of which hopefully each of one will have a single of these bits, and then they'll have the different fluorescences read out on the instrument. And so it's really that simple as just prepping your DNA and putting it through the machine, getting the cloud of values, and then modeling what these different clouds mean in terms of the mutation you're trying to detect. And usually here, this is a very targeted approach, meaning you always want to detect a specific mutation in a specific place in a specific gene per fluorophore per color. And you get plots like this that basically say how many of your molecules are wild type or how many of them are mutant, and in different colors for different mutations that you're targeting. NGS is different or has the potential of doing more, and here's in a slide that shows exome sequencing as a possible way of doing NGS of liquid biopsy samples and finding out if there are mutations in the exomic part of the genome. And the concept is the same. You're trying to identify mutations selected by treatment. There is also people out there doing whole genome sequencing of liquid biopsy samples, which is more costly, but it gives you a full profile. And some of these are preclinical, or let's say in discovery projects. And if you do it in a target approach, the concept is the same as we saw in the ddPCR slides. You're targeting by some hybridization method, specific regions that you want to sequence, and then you count how many times you find them mutated or in wild type. One important consideration, and there's more and more science being discovered through this, is if you want to find these somatic mutations at a given frequency and then associate them to the developing tumors in your body that shed DNA into the bloodstream, you have to take into account that some mutations that you find in your bloodstream are simply from the phenomenon called clonal hematopoiesis. And so if you take a blood sample and isolate the cfDNA, you've lost the white blood cells DNA where clonal hematopoiesis takes place, even though these cells themselves can shed DNA into the bloodstream, and you would find them in your cfDNA sample. So clonal hematopoiesis is related to age, and it basically works like this. At birth, you have a bunch of hematopoietic cells, and at middle age and in advanced age, some of them get damaged, and they create clones of cells that are all damaged for a specific damage. DNA damage, telomere attrition, epigenetic dysregulation, etc., etc. And it's been linked to atherosclerosis, cancer, and other conditions. This can happen also via extrinsic influences, and one of them is also, well, very much related to cancer, which is chemotherapy. So chemotherapy can kill your cancer or put your cancer at bay, but it also creates extra damage, which can accelerate the clonal hematopoietic effect, right? And recently in the ASCO meeting 2018, Charles Swanton, who has been doing a lot of work from his base in London, showed extensive data for what he calls CHIP, clonal hematopoiesis of indeterminate potential. And it shows how CHIP is related to age and how it's associated to cardiovascular disease, overall mortality, hematological malignancies. But importantly, because white blood cells contribute to the cfDNA fraction, CHIP can confound cfDNA-based assays in the sense that the white blood cells can also confound circulating tumor cell or tumor tissue sequencing assays. And so what they did with this project that they call the CCGA, it's a counterpart of the TCGA, but for cfDNA, is to take about 15,000 samples from individuals, blood samples, tissue samples, cancer only, and then follow-ups. And then for the blood samples, apply a collection of assays, targeted sequencing, both of the cfDNA and the white blood cells, whole genome sequencing, both of the cDNA and white blood cells, targeted and whole genome bisulfite sequencing of the cfDNA to find out about the epigenetic state of the individuals, and whole transcriptome sequencing of the cfRNA in this case. And for the tissue samples, they did whole genome sequencing of the tumor sample. And then they did follow-ups and tried to understand how the cancer progressed. In the targeted sequencing, both for the cfDNA and the paired white blood cells, they did 507 gene panel, and they aimed at 60,000 depth, find variants from the white blood cells, and then do a final filtering for non-synonymous SNBs and indels. And so from previous studies, people said, okay, if I got variants at low variant allele frequency, are they all going to be cancer? And the question is, how can I filter out the ones that, even though they're at low frequency, are not actually cancer? Doing ultra-deep sequencing in this CCGA study, they were able to find about 10% of white blood cell match variants at around 1% VAF. And so correlating those in the cfDNA and the white blood cells, there is a very nice correlation. Well, nice in the sense that you can find it, but not nice in the sense of what it affects the diagnostic of this, because it can be a confounding signal in cfDNA-based assays. And CHIP increases with age and has similar prevalence in cancer and non-cancer patients, meaning if you have a healthy individual, every 10 years, there's going to be an increase of 160, 170% in the number of CHIP variants. And so any healthy individual of certain age will have what looks like somatic mutations that could be associated to cancer, even if it's a healthy individual. Where are these mutations? If they weren't in any cancer genes, then it would be fine, but they also are in cancer genes, as shown here. And this suggests that the screening population may have confounding CHIP variants in their cfDNA that must be accounted for. Otherwise, you're screening healthy individuals that just have clonal hematopoiesis happening due to age, and you may consider them as cancer patients. And nearly all these white blood cell match variants in cfDNA are specific to individual patients in cohorts, right? So these what's called private mutations only seen in one individual suggests that targeted approaches will require the matching of white blood cells to correct for this false positive identification of variants. One way of filtering this is to use natural selection methods or molecular evolution methods, and this is an adaptation of Martin Corena, a paper in Cell in 2017 that shows that positive selective pressure results in more driver mutations, and these can be measured by their what used to be called DNDS score, the ratio of synonymous, non-synonymous, and synonymous mutations. And so one way of filtering out mutations that are not driving cancer from the ones that are driving cancer is to apply this kind of methodology, which was pioneered by Martin Corena. And so if you apply this technology in this methodology in samples in this CCGA study, also this from Martin Corena Cell 2017, you can find plenty of genes that are driving cancer, and some of these also have strong positive selection for clonal hematopoiesis. So because of clonal hematopoiesis, this is one aspect of the work of doing liquid biopsy NGS that if you're not aware of it, then you may be generating lots of false positives, and it's an example of how complicated the field is, in the sense that you have to be aware of everything, all the biology that's happening inside the blood, to be able to have an accurate diagnosis of cancer through liquid biopsy. Understanding biological relevance of DNA in blood allows us to calibrate the expectations of liquid biopsy assays. This is an evolving field with active new development, so we'll see more and a wider offering in diagnostic assays using liquid biopsy. And then the remaining question is there going to be a one-size-fits-all assay for all types of cancer that are performed by a liquid biopsy, or are there going to be tailored approaches to either diagnose, the diagnosis, prognosis, or treatment monitoring of cancer? It remains to be answered. So follow me on Twitter if you want to continue hearing about the Omics cast and the episodes that I'm going to be publishing. And you can contact me on LinkedIn, ask me questions there. I'm happy to answer any questions you have. And a reminder that this slide deck is available upon request. I'll see you in the next episode of the Omics Cast.